Okay guys, what we are actually going to be doing is we are going to be looking at our genes and chromosomes kiosk, and I am going to be recording the benchmark for that. So the first thing that we're going to talk about is what our genes and chromosomes kiosk is about, and we're really going to be talking about how DNA is organized. So the first thing that we're going to talk about in reference to the DNA being organized is about just how big DNA is, right? The idea that DNA is coiled and coiled and coiled and coiled, so tightly wound up inside of the nucleus of a cell that you never actually get the full effect of just how long DNA actually is. So the idea that if you lined up all of your DNA in all of your cells, it would actually reach to the moon and back 3,000 times. That's pretty long when you think about how teeny tiny your cells are and how tightly wound that DNA must be to fit in that space. So that's a whole lot of genetic material, all right? But not everything that's in there actually has any sort of purpose at all. There's only a small chunk of DNA that actually codes for important information. All right, that important information or the important chunks of DNA are referred to as being genes. So, a gene is going to be a sequence of DNA that codes for a certain trait. So the idea that if we had our strand of DNA, here's our lovely strand. I know how much you guys enjoy my fantastic drawings. We got our bases in the middle, right, making up the rungs of the ladder. You perhaps may have a segment like this that codes for a gene. That may just be a chunk of DNA coding for nothing of relevance. Whereas from here down, maybe another gene, right? So the genes are kind of interspersed throughout all of the nonsense, the unimportant information that's in the DNA segment. Right? So there's thousands of genes, 20 to 25,000 in the entire human genome. And every single gene is 10 to 15,000 bases. That's a whole lot of bases. It's approximated that there are 3 billion base pairs in human DNA. It's a whole lot of genetic information for us to be looking into. Right? So, approximately 5.5 feet of that DNA gets coiled up into each of your individual chromosomes. Right? You guys have heard the term chromosome when you're talking about genetic information. A chromosome really is just made of DNA. But more specifically, it's DNA wound up around proteins. All right? So we're focusing on the idea that it's DNA plus those proteins. All right? When we talked about our DNA extraction lab, all right, we talked about the fact that DNA winds itself up, and inside of those winded points, there are these little proteins. Not sure if you guys really necessarily remember, but those little proteins are something that are referred to as histones. Right? So histones are the proteins that DNA winds up around. So a chromosome, right, another way to think about it is that a chromosome equals DNA plus histones. Right? When you take those two things and you put them together and you wind them up really, really tightly like they're supposed to be, then we call it a chromosome. And DNA, they're like little pieces, the histones are like little parts of glue holding the DNA together. They're like balls of glue. So this picture right here is just kind of showing you that the DNA is going to wind up around those histones. All right? And here we show the DNA winding up around histones. All right? Then it's going to continue to wind up around itself. All right? It's going to condense. The more it winds and the more it winds, it's like the idea of string. The more you twist it and twirl it and twist it and twirl it and twist it and twirl it, it starts to curl up on itself. That's what DNA does. It just keeps coiling so that it starts to wind up on itself until the point where you get these things, these chromosomes. It's so thickly coiled that it's no longer this little teeny tiny strand. Right? So when it finally gets really wound up, we refer to it as being a chromosome. Now, when we talk about parts of a chromosome, typically you're going to see a chromosome as being one single chromosome with gene bands. This should probably be somewhat familiar with you guys. The idea of the banding should be familiar from the DNA fingerprinting gizmo that you guys did. So typically what you're going to see are going to be single chromosomes. But every now and then, you may come across an image that refers to this structure. Each side of this would be one individual strand of DNA coiled up around histones. Another strand of DNA coiled up around histones. All right, these two parts are going to meet and join together in a point called the centromere. The only time that you're ever going to see this, all right, it's only in this form during cellular division. But it's important for you guys to learn this because this is our fundamentals of genetics unit, and cellular division is going to be one of the next things that we cover. So it's important for us to get these vocabulary words out of the way. Typically, I would call this a chromosome and this a chromosome. But when it's joined together like this, the whole structure itself is a chromosome. Each individual one gets called a chromatid. So, chromatid is half of a chromosome in this structure, all right? It takes two chromatids to make up a chromosome, all right? But the important thing for you guys to also realize is that chromatids are identical copies. We sometimes refer to them as being sister chromatids, right? And by identical copies, you have an original chromosome and its copy. So in cell division, it's important to copy your genetic information because when the cell divides, you want each new cell to have a full set of genetic information. You don't want to forget to copy a chromosome and for the new cell to be missing that chromosome. All right? So it's important to have copies. So in this case, we'd have an original and we'd have the copy. But the only time they're ever going to join together like this is during cell division. And that centromere, that middle point, where the sister chromatids are going to attach. But, like I said, typically when we refer to chromosomes, that's what we're referring to. This is just another picture showing you about how the DNA winds itself up. All right, if you look here, we've got those histones all tightly wound up together.